First speaker is Cody Champion from New Mexico State University, so all yours. So, laser forward barbers.
All right. So hello, my name is Cody Champion. I'm from New Mexico State University working in Dr. Shu's lab. And what I'm going to be presenting is NADPH cycling inhibition impedes fecundity in Anopheles gambiae. So this is a different title than was posted in the program. What we're doing is we're looking at the total NADPH levels being reduced using inhibitors as well as paraquat in mosquitoes and looking at various life traits. So I'm sure most of you know the life cycle of the mosquito, but just to make sure everybody's on the same page, the eggs are laid, and within 2 to 4 days, they hatch into larvae. The larvae will then go into pupae, and these are all water-based organisms. And then the mosquitoes will then emerge and live for 2 to 6 weeks. And what is important for our study is that mosquitoes will either feed on sugar meals or they'll feed on blood meals. Males will only feed on the sugar, whereas females will feed on blood in order to obtain protein and iron for egg production. And that leads us into redox homeostasis. So when a mosquito takes a blood meal, it's going to be ingesting hemoglobin, which will then produce oxidative stress, and I'll talk about that in a minute. Also oxidative metabolism, so both males and females will undergo this as well. So when the electrons are being passed through the electron transport chain, if oxygen comes in and interacts with any of the first two complexes of the electron transport chain, that oxygen then can become a free radical due to its unpaired electron. Also, oxidative stress can be beneficial for mosquitoes. So for immune responses, mosquitoes can mount a defense against mosquitoes using the IMD or toll-like pathways and then an NADPH-dependent dual-oxidases to produce oxidative stress within the midgut, which will then clear bacteria. So when heme is ingested by mosquitoes, the heme will be digested into two components. The first one being the biliverdin derivatives, and then the second being the iron species. So this iron species can then go on and produce oxidative stress through the Fenton reaction. So what that does is the Fenton reaction essentially will complex with the hydrogen peroxide, which is a very moderate reactive oxygen species. It doesn't produce as much damage as a hydroxide radical. The hydroxide radical is one of the most heavily damaging free radicals in biological systems. What happens when this free radical interacts with various biomolecules is that they become oxidized. So particularly lipids, proteins, nucleic acids will all become oxidized, and when a biomolecule is oxidized, it loses its functionality. So when mosquitoes do encounter this iron species, they do have systems to excrete it. So this iron is either used to create eggs, used in the synthesis of eggs, or is excreted. Over 90% of this is excreted from the mosquitoes, and very little, about 2%, is used within the eggs. So once a blood meal is taken, just kind of summing up the entire model that we have, is the iron is the primary driver of oxidative stress within mosquitoes. So that undergoes Fenton's reaction and all that chemistry and produces oxidative stress. And then the mosquito has two ways to deal with this. It can either use inherent systems within the mosquito itself, or it can rely on its microbial community to help mitigate this oxidative stress. And actually, one of the students that we have in the audience from our lab has shown that specific bacteria found within the mosquito midgut have different tolerances for oxidative stress. So it appears that there is some type of interaction between the mosquito's midgut microbial community and the mosquito itself. For my talk, I'm focusing on the global system, so using inhibitors that will reduce NADPH or induce oxidative stress on a global level, so not specific to the mosquito or to the microbial community, but rather to the organism as a whole. So when oxidative stress is encountered, there are inherent systems to deal with this stress. So you have superoxide dismutases that will help alleviate superoxide ions, and catalase, which will then remove hydrogen peroxide. And then also you have antioxidants such as glutathione, which can then be recycled using NADPH, using glutathione reductases and glutathione transferases as well to help with other processes in the cell. And what we're focusing on is the source of power for this NADPH production. So NADPH is primarily created through the pentose phosphate pathway. So this is a pathway that is an alternative pathway to glycolysis. So glucose, when ingested, will go to glucose 6-phosphate, and they can either go through glycolysis or through the pentose phosphate pathway. And there are several steps and mechanisms by which this pathway can be modulated. So for instance, pyruvic kinase, if oxidized, will become less active, thereby creating more substrates within the glycolysis pathway, and then pushing the pressure into the biosynthesis of the pentose phosphate pathway, producing NADPH. Also, there's various shunts such as TPI that can create more or less substrates within the glycolysis. There also are other pathways that can produce NADPH, such as malic acid dehydrogenase and isocitrate dehydrogenase. So our data that supports this assertion that the pentose phosphate pathway NADPH production is important to the mosquito is first supported by a transcriptomics experiment that we looked at. So when mosquitoes take a blood meal, as shown in the blood, this is 24 hours post-blood meal, and the blue is just this baseline sugar feeding only. We see that GAPDH, which is involved in the glycolysis, is increased in the sugar fed, which makes sense. They're being primarily fed on sugar. They aren't being exposed to protein or anything else. And also the TPI, which is a metabolic shunt pathway that will create different substrates within the glycolysis that will allow more substrates to flow through glycolysis without backing up the system and pushing it into the pentose phosphate pathway. We see that these enzymes are decreased after blood feeding, which this supports the idea that the pentose phosphate pathway is being increased. However, there's an alternative explanation that just because they fed on blood, they're taking less sugar, therefore glycolysis and the pentose phosphate pathway are not being utilized within the organism. But this is a good starting point for us to look at this, in that there is some changes in glycolysis and the pentose phosphate pathway after blood feeding. The next experiment that we did is we did a metabolomics experiment. So what this is an untargeted metabolomics experiment. So we pooled 50 females, homogenized the midguts, and then we had a metabolite profiling with this one. So this just found the relative abundances of each metabolite within the system. We then used pathway enrichment analysis, so the POPI package from R. And what this does is it provides a metabolic flux score for the different pathways within the mosquito. So there are many pathways that are changed before and after blood feeding. So we looked at sugar-fed only, 24 hours post-blood meal, 48 hours post-blood meal. And what we find is that the pentose phosphate pathway, so those metabolites that are associated with the pentose phosphate pathway, not with other pathways such as glycolysis, are increased, so relative to the amount of other metabolites within the system, 24 hours post-blood meal. We see it return back down to relatively close to baseline levels, 48 hours post-blood meal. So this is more concrete evidence that the pentose phosphate pathway is contributing to the digestion of blood, and most likely contributing by providing NADPH production within the cell. So this is looking at the actual data that we use, so without the metabolite abundance enrichment analysis. Glucose 6-phosphate, 6-phosphate glucoalactinate are all higher in the blood feeding, so that's what these, what these boxes here show. The gray boxes are the sugar-fed, so you can see in the sugar-fed groups that they're lower in metabolite levels versus when they're blood-fed. Additionally, when I mentioned that there's antioxidant that can help prevent oxidative stress damage, the ones that we're looking at specifically are glutathiones, so glutathione oxidized form, which is GSSG, and then the reduced form, so GSH. So the GSH is the form that is able to actually respond and mitigate oxidative stress, whereas the GSSG form is the spent form. And what we find is that the oxidized form is higher in post-blood meal-fed mosquitoes when you compare it to the sugar-fed alone. So this is our sugar-fed, low, almost no detectable levels, higher levels in the blood-fed. So in order to probe this, we did some feeding experiments. So we fed either lycopene, which is our antioxidant control, so this should be able to mitigate oxidative damage by us adding to the system. The second is 6-amino-nicotinamine, which is an inhibitor of the pentose phosphate pathway, as well as other NADPH production pathways. So that's why we're hesitant to say that that the pentose phosphate pathway is the sole pathway for NADP production within the mosquitoes, but we are designing experiments that will help us piece this apart. We use paraquat, which is a commercial herbicide that also induces oxidative stress. And then finally, we added NADPH to help us confirm that 6-AN's effects were due to NADPH production repression. So as I mentioned before, that 6-AN is a pentose phosphate pathway inhibitor. It's the most widely known inhibitor for the pentose phosphate pathway, but it is a competitive inhibitor that mimics NADP. So it's difficult to say that we're inhibiting all of the NADPH production only through the pentose phosphate pathway, but rather we're just trying to inhibit NADPH production globally. Paraquat produces oxidative stress through an NADPH-dependent mechanism. So when paraquat will complex with NADPH, it'll then form a paraquat radical, and then that paraquat radical, when it complexes with oxygen, will then form a reactive oxygen species. So what we find when we feed paraquat to mosquitoes is that paraquat will reduce the number of eggs that mosquitoes will produce. So these are dissected out eggs. So we did look at their ability to lay eggs. So there's no differences in the mosquitoes that were fed paraquat versus sugar in their ability to lay the number of eggs versus otherwise. And then we see that lycopene has no effect when you feed it to the mosquitoes by itself. But when you feed paraquat plus lycopene, we see that a return to normal baseline for egg number. So this tells us that paraquat's mechanism for reduction of egg number is through oxidative damage. The next experiment that we did is we looked at feeding 6-AN, so inhibiting NADPH production. And what we find is that 6-AN lowers egg production. However, when we feed lycopene, which will reduce oxidative stress, we see it turning to baseline. When we feed NADPH, we see no difference. And then finally, when we feed 6-AN plus NADPH, we see a return to baseline. So this tells us that the lowering in fecundity is most likely due to oxidative damage because lycopene was able to reduce it, and also due to NADPH production repression because when we add exogenous NADPH, we see a return to baseline. Finally, what we did is we looked at the biochemical environment within the mosquitoes. So this is a strip plot showing the distribution of egg number within normal sugar-fed mosquitoes. So the red represents low egg producers, the yellow represents high, and green yellow represents middle, green represents high egg producers. And what we did is we measured NADPH, NADP, and oxidized versus glutathione levels. So right now we have oxidized versus reduced glutathione. So the higher this number, the more oxidatively stressed the system is. And what we find is that sugar-fed mosquitoes have a ratio that isn't damaging to them. However, when we feed 6-AN, we see an increase in this ratio. So that means that there's more NADP versus NADPH, which is the active form of NADPH. And so this supports our assertion that NADPH is vital for the regeneration of glutathione. Secondly, we then looked at NADPH levels versus NADP. And what we find is that egg layers that produce a low amount of eggs have unfavorable ratios of this. So they have high amounts of NADP versus NADPH. And 6-AN, this doesn't tell the complete story because when you look at these individually, you see that they're just below our baseline measurements. So 6-AN has almost completely repressed the production of NADP and also NADPH. So this may be due to metabolic collapse. We're not quite sure yet on that. Finally, the conclusions that we can draw from this is that oxidative stress limits fecundity. 6-AN inhibits NADPH production, most likely through the pentose phosphate pathway, but not necessarily. Decreasing NADPH levels leads to lower fecundity, and this could be due to oxidative damage or by reducing the amount of NADPH that can be used for biosynthesis. And we have the, running out of time here, applications is that we think that we may be able to use this as an adjuvant to existing pesticides to help them increase their viability. And finally, I just want to thank my lab members, Dr. Shu's lab, my committee members, and our collaborator, Dr. Shirley Luckhart, who helped us with metabolomics. And thank you.
Thank you, Cody. We have time for a question. One question, if anybody wants to ask a question. Yes, no. Well, moving.